introduce today's speaker? Okay, today's speaker is Emilia Huerta Sanchez, who's a postdoc in Rasmus Nielsen's lab and soon to be an assistant professor at UC Merced, starting in January. She received her PhD in applied mathematics from Cornell, and then spent a year teaching in the statistics department at UC Berkeley before being a postdoc here in Rasmus's group. And today she's going to speak about high altitude adaptation, an example of adaptive introgression in humans. So Emilia, thank you.
Thank you, Michael, and thank you for letting me invite myself to give the seminar. As Michael mentioned, I am soon to be at UC Merced, and I'll be here till the rest of the semester. So if anybody wants to speak with me before I leave, you can find me in my office upstairs in the Nielsen lab. In the Nielsen lab. Okay. And so today I'm going to talk about some of the work that I have done here with Rasmus, but first, very broadly, let me tell you what's one of the major things that I'm interested in, and that's understanding how humans have adapted to different environments. And so we know that humans originated in Africa, but they left Africa and they moved around. They went to other places within Africa and outside of Africa. And in doing so, they encounter changes in temperature, changes in diet, changes in pathogen exposure, and changes in altitude. And so these are likely to have acted as selective agents that led to local adaptations. And so the way I like to address these questions, or the way that I have been addressing these questions up to now, is by combining a few approaches. And so by that I mean that I like to look at mathematical models of evolutionary processes to compute expectations, that we can then fit those expectations to the data using a statistical framework. But of course the data is never pretty, it's dirty, and so you always have to come up with clever and robust ways to data processing techniques that accurately estimate genetic variation. And so today I'm going to tell you mostly about one of the projects that I've done a lot of work on, which is adaptation to high altitude. And I started this project back towards the end of 2009. And so I went into Rasmus's office, and I had a meeting with him and another fellow postdoc called Nicolas. And he still called Nicolas. We were having a meeting because we had been analyzing the data on 200 Danish exomes. And at that meeting, towards the end of the meeting, he said, well, our collaborators have sequenced 50 Tibetan exomes, and they want to know what are the genes responsible for this adaptation. And he said, do you want to work on it? And I immediately said yes, and I was very excited. And the reason I was really excited is that for the 200 Danish exomes, I had actually done my first positive selection scan using those data. And at the end, you come up with a list of genes, and then you have to figure out, like, okay, why are these genes interesting? What's the biology behind this, and what selection pressure are we talking about? And so I remember giving the top genes to Nicolas, and he did a lot of analyses. He did Go analyses, and it was still difficult to come up with why were these genes selected. But this project was different, and it was different because we knew what was at least one of the selective pressures, and that was the lack of or the reduction of oxygen. And so just to give you an idea of how what the difference is that in some of these humans live in regions that are more than 4,000 meters in altitude, and in those regions, for every breath of air that you take, there's about 40% fewer oxygen molecules than at sea level, and so that's a huge reduction. And furthermore, there were a bunch of other studies that had already done a lot of work in these populations trying to characterize whether or not this was a genetic adaptation or whether it was just these populations being acclimated to that high altitude. And so one of the studies looked at Tibetan populations here on the left and Han populations on the right. And the Tibetan are high altitude natives, and the Han are low altitude natives. And what they measured was hemoglobin concentration here on the y-axis and altitude on the x-axis. And what they found was that if you look at the Han, as altitude increases, so does hemoglobin concentration. And when you look at the Tibetans, as altitude increases, hemoglobin increases a little bit, but not nearly as much as in the Han. And so you can see here that the response is actually quite different between these two low and high altitude populations. And so that was one indication that the Tibetans had a different mechanism to deal with the lack of oxygen. And another interesting finding was measuring birth weights. And here, what researchers did, they look at different altitudes and they measure birth weights. And what they found was that individuals of high altitude ancestry had babies that were heavier than individuals of low altitude ancestry. And so these two, this and the previous findings suggested that this was a genetic adaptation. And this is what they had actually speculated all along. And so here we were. We had 50 Tibetan exomes, and we wanted to see whether we could actually identify the genes responsible for this adaptation. And so after doing a lot of statistical tinkering with the data, we could actually estimate allele frequencies for the Tibetan exomes and also for some Han sequencing data. And at the end, one way to summarize the whole data is to look at the two-dimensional side frequency spectrum. And I think many of you have seen this figure maybe, but I'll go over it again. So here, on the x-axis, we have the Tibetan frequencies, and on the y-axis, we have the Han allele frequencies. And the different colors are just the different heights of the bins, and this is really just a histogram of the frequencies of the mutations. And so what you can see here is that most of the mass is on the diagonal, so most of these, so the Han and the Tibetan population have pretty correlated allele frequencies. There are some mutations that are at high frequency in Tibet and a low frequency in the Han. But we wanted to have a measurement for every single gene in the genome. And so to do that, we assumed that the relationship between these three populations, and by that I mean the high altitude population, which are the Tibetans, the low altitude population, and the outgroup, we had Danish population. And the reason for using an outgroup was that if there was a large frequency difference between the Tibetan and the Han populations, that could be due to selection in the Han or selection in the Tibetans. And so to focus on the unique changes in the Tibetan population, we could use an outgroup. And essentially it boils down to estimating this branch length, and you can do that by estimating these T's, and these T's are just the transformation of population, a measurement of population differentiation. And so the idea is that if a gene is neutrally evolving, then you would expect that the high altitude branch would be similar to the low altitude branch because these populations are closely related. But if there's selection acting on a gene, then you would expect this branch to be larger because at that gene, the low and the high altitude population would be very differentiated. Okay, and so if you do that for every single gene, you find that E+1, that gene has the largest value for population branch statistic. And when you do that, and so we found this gene, and we were really happy because this gene is also called the hypoxia-inducible factor 2, and you can't do better than find a gene that has hypoxia in its name. And so we were happy because it made a lot of biological sense. It regulates the response to hypoxia. It also is involved in the red blood cell production and other very things that make biological sense. And interestingly, the two mutations that were driving the signal in this gene are in an intron. And so, which was interesting. But if you go back to the two-dimensional side frequency spectrum, you find that these two mutations that have the largest frequency difference, where Tibetans have high frequency and the Han have low frequency, are those two mutations in that gene. And so one of those mutations, this one, has a very large frequency difference between the Tibetan and the Han. And that's actually quite large for humans because these two populations are so closely related. And EPAS1 was not the only gene that we identified. We also identified other genes that made biological sense, such as EGLM1, which is also in the hypoxic pathway, and also the hemoglobin proteins, which also make biological sense since they bind oxygen. But if you take that mutation in EPAS1, it has the largest frequency difference, and if you genotype it in more than, we genotyped it in more than 300 Tibetans, and we also collected hemoglobin measurements, you find that the individuals with homozygous for the derived allele have a lower hemoglobin concentration, and this association is significant. And actually that makes sense from what I told you earlier, that Tibetans don't actually increase hemoglobin concentration. And so after the end of the study we had more questions. Well, we wanted to know whether we could actually estimate the timing and the strength of selection. But of course, if you want to, the reason for that is that we had EPAS1 and then we had these other genes, and we wanted to know what was the series of evolutionary events that took place. Were the genes selected at the same time, or were they selected at different times? But of course, if you want to estimate parameters, then you need to have a model. And so the models that most of the people in the field were talking about were selection on a de novo mutation or selection on standing variation. Whether or not a population has to wait for a de novo mutation to arise, or whether a population has enough neutral variation that if the environment changes, some of those mutations will become beneficial. And so we set out to first test whether selection on standing variation or selection on a de novo mutation was responsible for this adaptation. And so because we had found these two intronic mutations, we wanted to resequence the whole gene, because before that we only had exons and a little bit of intronic regions. And first of all, we wanted to confirm that indeed EPAS1 had a strong signature of positive selection. And so we sequenced the whole gene, and we sequenced it at a very high coverage in 40 Tibetans and 40 Han individuals. And so the first thing you can do with this data is to look at population differentiation, which is what I'm showing you here. So on the x-axis, I have the position of the gene, and on the y-axis, it's the measure of population differentiation between Tibet and Han. And what you can see here is that there's a bunch of SNPs that have very large frequency differences in these two populations. And just to give you an idea, genome-wide FSC between these two populations is roughly 0.02, and some of these SNPs have like 0.8 as a measure of population differentiation, which is really, really large. And so as I mentioned, we wanted to distinguish between these two models. So Benjamin Peter, an excellent graduate student at the time, he developed a method, approximate Bayesian computation framework, and he used information or summaries of the data that were summarizing either something about haplotypes or something about the frequency of the genetic variation. And so, and this method worked well, but when we applied it to this gene, we couldn't really say with confidence whether it was one or the other. We could definitely say that it was not neutral. Neutrality wasn't playing a role here, but and definitely selection on standing variation had more higher probability, but there was still a lot of uncertainty. And so at first we thought that maybe it was because we were not using a summary of the data such as population differentiation. But in fact, when I added this extra information, we still could not get a satisfying answer. And so, so that was depressing. We could get a bunch of interesting results using genomic amounts of data, but we couldn't say something interesting about this one gene. And so, so we went back to look at the data. And so when you look at this figure, you say, okay, there's a lot of mutations that have a large FST. That means there's a lot of mutations that have a large frequency difference. And when you actually look at the frequencies, you find that in some of those mutations actually have zero frequency in the hand. And so we decided to look at the haplotypes. But before I show you the haplotypes, so basically, after figuring out that we couldn't say something about this gene, there were two interesting plots that we had, that one and this one. And in this one, we were able to show that the signature in this gene could really be due to a local adaptation. And the reason for that is that definitely there was a lot of differentiation between the Tibetan and the Han. And you could ask, well, what is it with respect to other populations? Do we see that in other populations? Do we see such a striking signal where you have two closely related populations that have mutations in a gene that are very differentiated? And the fact is that only a few other genes, like the skin pigmentation genes, show such a pattern. Okay, so if you look at the haplotypes, though, this is what you get. And so when we saw this figure, we sort of had an aha moment, like, oh, that's why we couldn't get it. But first, let me tell you what this figure says. So each row here is a haplotype, and the first 80 rows are the Han haplotypes, and the top 80 rows are the Tibetan haplotypes. And if it's dark, that means that it's, in each column is a SNP, and here I have removed rare SNPs in both populations, or high-frequency SNPs in both populations. And if it's dark, that means it's a derived allele, so we're using the chimp as the ancestral allele. And if it's gray, that means it's an ancestral allele. Okay? And so, and the rows at the top are just showing the sites that are the most differentiated between these two populations. And if you just focus on this 5-SNP haplotype here, what you find is that this 5-SNP haplotype, it's at high frequency in Tibet, but you do not see it at all in the Han. And so that was the first signal that this haplotype had to come from somewhere else, because there are 5 mutations in a small region of 2,500 bases that are at high frequency in Tibet, but not at all in the Han. And remember, these two populations are very closely related. And so, but first we need to figure out whether selection on standing variation or selection on the de novo mutation could actually explain this pattern. And we found out that no, we couldn't. And so just to give you some intuition of why that's the case, in a model of selection on a de novo mutation, you have your population of haplotypes, and you get a beneficial mutation in one haplotype. And so then what happens, that haplotype will rise to high frequency. And with it, it's going to take all of the linked neutral variation. But if you group the haplotypes with the selective mutation and those without the selective mutation, you find that there's still a lot of shared variation between those two haplotype classes. On the other hand, on a model of selection on standing variation, again, you might have the mutation, a neutral mutation that's at low frequency in your population, but let's say that due to a change in the environment, it becomes selected. So then that mutation will rise to high frequency, but this time there will be more diversity in it will not just be one haplotype going to high frequency, but more than one haplotype going to frequencies. But again, if you group the haplotype classes into those that have the beneficial mutation and those that don't, they'll still share a lot of that neutral variation. But if you have something where another population donates a haplotype that happens to be have a beneficial mutation, then this haplotype would be introduced into the population. This haplotype would go to high frequency, but now there's a lot of other neutral variation that's also new into the population. And so when you group the two haplotype classes, you will have a lot of variation there that's not in one haplotype class, but in the other. And so this is one of the reasons why we couldn't, either of those models couldn't really explain that pattern. And so now we knew that it was likely that this came from another population. So now I just wanted to look at this 5-SNP haplotype and ask, well, some other human population must have it. It can be that they don't. And so the obvious place to look is to look at the 1,000 genomes data, and when you say, okay, what are the haplotypes at those 5 SNPs, and what's the frequency of those haplotypes? And what you find is that the Tibetans have that 5 SNP haplotype at high frequency, and that's shown in green, but almost everybody else is yellow. They have the complete opposite haplotype. And there are only 2 Han individuals that are heterozygous, where one of their haplotype actually matches the Tibetan haplotype. But nobody else had this haplotype. And so that was really strange, because these are human populations. We share almost everything. So I thought that we would be able to find this haplotype somewhere else. And so I was talking to my office mate, Mehmet, and I said, Mehmet, what's going on? Like, this is really weird. And then he said, he looked at me very seriously and he said, it's the Yeti. And so I said, what? He said, so we had a good laugh. And then, but that gave me the idea to look at archaic humans. And so I ran over to see my friend Flora, who I knew had like access to all this data, and I said, can you just tell me what's variation at these 5 SNPs? And so she did that for me, and then we found that haplotype doesn't match the Neanderthal. It doesn't match most other humans, but it does match. It has a perfect match with the Denisovan. And so that was weird, because I said Denisovan. The only thing, so I didn't know very much about the Denisovan, and so I knew after doing a little bit of reading, I found out that the Denisovan is called the Denisovan because the bone was found in the Denisova cave in the Altai Mountains in Siberia. And I'm just wondering where the Tibetan plateau is for perspective. And so I knew that there were 2 bones found in the Denisova cave, and one of them was from a Denisovan, and that was published in 2012. And the other one was what they called the Altai Neanderthal. They couldn't call it Denisovan Neanderthal, so they called it the Altai Neanderthal. And the only thing I had heard about the Denisovan was that the population that had some Denisovan ancestry were the Papuans. And the first paper suggested there was they didn't find any evidence of a mixture in Southeast Asia. And so, but then I found another paper that suggested that they could say that there was likely to be some ancestry in Papuans. And I think now, in the new findings, everybody agrees that there is some Denisovan ancestry in East Asia. Okay. And so, and of course, I have shown you so far I've only told you stuff about 5 SNPs, but if you look at the whole region, which is a region of 32 kb, and if you look at the, I've placed the Denisovan haplotype at the type, you can actually see that it quite matches, it matches the Tibetan haplotype almost at every position. There's obviously a few positions that are different, but it's closest to the Denisovan haplotype than to anybody else. Okay, but of course here I'm just showing you 3 populations. I mean the Denisovan, Tibetan, and the Han. You can also ask, well, what is the haplotype in that whole region in the other individuals? And of course that's a lot of haplotypes. That's more than 2,000 haplotypes. But one way to visualize there's a lot of rare haplotypes. So one thing you can do is to just look at the 40 most common haplotypes, and that's what I'm showing you here. So every circle is a haplotype, and there's an edge between 2 haplotypes if they have the least number of differences, and the width of the edge represents the number of differences between the 2 haplotypes, and the size of the circle represents the frequency of this haplotype, and the pie charts are frequencies for each population. And what you find is that at the end there you see Here you see these are 2 Tibetan haplotypes, and only Tibetans have this haplotype. And what you can see is that the other closest haplotype is the Denisovan haplotype, and there's 9 differences between these 2 haplotypes. And if you ask what's the next closest haplotype, that's this haplotype here, but there's 40 differences between that haplotype and the Tibetan haplotype. So, but what's interesting is that the next closest modern human haplotype to the Denisovan haplotype is 35 mutations. There's 35 mutations in which they differ. So in fact, the Tibetan haplotype is so all the other modern human haplotypes are closer to the Tibetan than all other human haplotypes are to the Tibetan haplotype. And the reason for that is that, and that makes sense because the Denisovan lineage went extinct, but when that haplotype was introgressed into the ancestral population of Tibetans, it kept accumulating mutations all the way to the present. And so it makes sense that now modern humans are further away from the Tibetan haplotype than the Denisovan haplotype, because the Denisovan haplotype stopped accumulating mutations. And so this is one characteristic that you would expect to see if a region has been introgressed. But of course, this is a small region, so you also have to make sure that you have to rule out that it's not lineage sorting or shared ancestral variation. And so we did a lot of analyses to rule that out, and we managed to find this was still significant. Okay. Okay, but of course there's, I hope now you are convinced that this is a good example of adaptive introgression. And of course there's now more questions. We are always asked, well, do you think that the Denisovans were high altitude adapted? And so, I don't know, and we probably will never know. I think the closest we can get to knowing is that if we actually find a causal mutation in the Tibetans, maybe if the Denisovans have that mutation, then maybe we can speculate that maybe they were, but we don't know. And the next obvious question is, well, do Tibetans have more Denisovan ancestry than, say, other East Asian populations like the Han? And so Melinda, a fabulous grad student, has done some preliminary analysis, and that has shown that that's not the case. There's actually enough similar levels of Denisovan and mixture in Tibetans as there is in Han. And so what we think is that it's really, this haplotype was probably drifting in the ancestral population of the Tibetan population and the Han population, and then when Tibetans moved to high altitude, it became selected, and it was really selection that drove it to high frequency. And other questions are, well, when did the mixture occur? I was surprised that there's actually no estimate yet for when Denisovans introgressed into modern humans. There is an estimate for when Neanderthals mixed with modern humans, but I think it's going to change soon. I know David Wright has access to a lot of Papuan genomes now, so it should give us some estimate soon. And what about the other genes? Are they adaptively introgressed as well? The other genes that seem to be under positive selection. And of course you have to, but to answer these questions, we really need to model both selection and introgression jointly to understand this adaptation. And Amy Ko, actually, she's another fabulous grad student. She has developed a hidden Markov model to actually infer Denisovan ancestry. And so we can apply this hidden Markov model to this example to actually get the distribution of track lengths and see if we can use that distribution to get some of these estimates. And of course, more generally, we want to know what is the relative contribution of adaptive introgression in human evolution, and what other nice gifts did we get from archaic humans? And so I think, so right now we're scanning the literature, and Fernando Racimo, another superb grad student, is helping me with this, and he made this nice table. So these are some of the examples that have been proposed to be adaptively introgressed, and so but the approaches that people have taken is either to detect signatures of selection in regions previously known to be introgressed, or to detect signatures of introgression in regions known to be selective. And so we really need to develop methods that can jointly infer the probability of a mixture and selection in a particular region. And of course, this is not trivial because we have to be able to, if you have a small region, then we need to be able to distinguish that this is not just shared ancestral genetic variation. And we can just also, if you have an introgressed region, we need to recalibrate because if you just use summary statistics that are known to detect positive selection, that's assuming a model that doesn't include introgression. And so we need to be careful with that as well. And so with that, I would like to thank Benjamin, Nicolas, Mehmet, John, Rasmus, and members of the Nielsen and Slacker Labs, our collaborators at UGI, funding from NSF and NIH, and I am looking for postdocs and grad students. So if anybody's interested or you know someone that's interested, please tell them about me. Thank you.
So I was just curious to know these other high elevation peoples in Ethiopia or in the Andes, what kind of parallel work is going on with those that would parallel the Han Tibetan system? And for example, since the Native Americans came over from Asia, did they carry Denisovan, haplotypes with them?
Yes, so they have found archaic ancestry in Native American populations. And there is work going on in the Andes and also in Ethiopia to find the genetic basis as well. And for Ethiopians, which is another population that I have worked in, we found different genes. They're in the same pathway, but they're different. And in the Andes, the only gene that's similar to the ones in Tibet is another gene called HGLN1. So there are that's the only gene, but it's still not clear what's going on there yet, I think. So, but yeah, there's work.
How high are the Altai Mountains? And what's the elevation of the cave where the
So the
Denisovan was
Altai
from?
Mountains they have plateaus at like 2,000 meters, which is a little bit high, but not super high. And then the cave, I think it's only like 300 meters.
Too bad.
Well, one thing that the Denisovians and Tibetans would share, but not with Papuans, of course, would be cold. How about cold adaptation?
Cold?
Cold.
Yeah. So I'm sure there's other things going on, but I mean, we haven't focused on those other selective pressures.
So when I look at people, and I've only been to the high Andes once, but what struck me was everybody was all bundled up in wools.
Yeah, so I'm sure there's like multiple things going on at the same time. Yeah.
And the somatotype for high elevation people is quite different than it is for low elevation people.
So Emilia, in your Tibetan haplotype data, so not all Tibetans have that characteristic haplotype, right, of the E+1 mutation. So do you know the phenotypes of those other Tibetans? Like, do they deal with altitude differently?
Yeah, we do have hemoglobin measurements, but I mean it's only a small sample of 40 Tibetans, so it's really difficult to do an association. But
So you don't see a striking difference between the ones that have the haplotype and the ones that don't?
nothing that was significant.
Okay.
If you look at in the initial study, the sites that show a big frequency difference between Denisovan and Han, other than EPAS1, do any of those others correspond to Denisovan haplotypes?
You mean other mutations?
So if you look between the Tibetans and the Han, so you have the plot of allele frequency differences, and EPAS1 is down here in the corner. But there are other things that are sort of at the extremes. Do any of those other ones correspond to Denisovan haplotypes? Have you looked at?
No, we haven't done like a full genome-wide scan to see what's, like if there's a region that, or a local search for Denisovan ancestry and whether or not they correspond to high frequency regions or differentiated regions. Because first we need to, but we're going to use the HMM to do local ancestry estimates, and then we can see that whether those are enriched for highly differentiated cells. But we haven't done that.
I mean, if the Denisovans were adapted to high altitude, you might expect the Tibetans are just reusing whatever's available that comes from them.
Yeah, yeah. But there are a couple of new mutations on that haplotype, so if it's those mutations that are selected or causal, then it could just be that they happen to land on this Denisovan haplotype. Or, I mean, we only have one Denisovan, it could be that, I mean, we're claiming that it's a population that's closely related to Denisovan, so since we just have one sample, it could be that somebody who we didn't sequence had that beneficial mutation as well.
So this is more of a background sort of question, I guess, a little bit off from what you were talking about. I mean, it seems like this mutation in this particular, or these mutations in this particular gene are essentially turning off an adaptive sort of a plastic response that is in fact negative. Like, so if I went to high elevation in the Tibetan Plateau, my hemoglobin will go up, and that's actually maybe not really a good thing, although it would allow me to breathe a little better. Is there an alternative is there an alternative gene or candidate gene that seems to be associated with improved performance? Because something else must be going on that allows them to be adapted.
Yeah. Yeah, definitely something else is happening, but we don't know what that something else is. And I'm sure people are, I mean, we don't even know what the causal mutation is. So some of these are in introns, so we want to do some functional analysis work to see whether we can do something with gene expression or look at relevant tissues like the placenta to see if we can find something interesting there. But we don't know what's being targeted by selection, what's happening.
Kind of along those lines, have you compared fetal hemoglobin in like Tibetans because they have higher birth weight babies?
Yeah, so one of those genes, one of them is fetal hemoglobin, and so yeah, it could be that selection is targeting something something like that just Tibetan women are better at having babies at high altitude, something along those lines, but we don't know. I know there are some groups collecting other phenotypes related to reproductive outcomes that may shed some light in that direction.
So on broader phylogenetic scales, I've seen some work that suggests that coding mutations in the hemoglobin gene are important for altitude adaptation across vertebrates. This is work by J. Storrs. So do you think it's the less deep split between the populations that you're studying that makes it more likely that you'll see regulatory compared to coding changes?
Yeah, probably. Yeah, I would say that's.
Because you didn't get any coding changes?
Yeah, we don't see any coding changes. Yeah. I can't remember whether this is a really conserved region or not.
Great. Well, you're here until the end of December, is that right?
Yeah.
And then off to UC Merced. Well, thank you very much.